Imagine living in a world where we can manipulate the building blocks of life itself. How would it change our lives? This is the world of biotechnology, a realm where science fiction meets reality, where we can engineer life to our whims. Biotechnology is the use of living systems and organisms to develop or make products. The field has expanded rapidly in recent years and has impacted everything from agriculture and medicine to environmental conservation. But how does it work? It all starts with understanding and manipulating life at the most fundamental levels. First up is genetic engineering, or the modification of an organism's genetic composition. Genetic engineering plays with the blueprint of life, our DNA. Scientists can add, remove, or alter genetic material, effectively designing organisms to perform certain tasks or exhibit specific traits. Imagine a bacteria that can eat oil spills, or crops that can survive in harsh climates. That's the power of genetic engineering. Next, we dive into the world of recombinant DNA technology. This technique involves combining DNA from different sources to create new genetic sequences. Think of it as a molecular cut-and-paste job, where scientists can combine traits from different organisms into one. This has been crucial in developing genetically modified organisms and has a wide range of applications from medicine to agriculture. Then we have DNA sequencing. This is the process of determining the exact order of the bases in a strand of DNA. It's like reading the genetic code, providing an insight into the organism's genetic makeup. DNA sequencing has revolutionized our understanding of life and has been instrumental in diagnosing and treating genetic diseases. Next in line is protein expression, which refers to the way in which proteins are synthesized, modified, and regulated in living organisms. Proteins are the workhorses of the cell, carrying out most of the biological functions. By understanding and manipulating protein expression, scientists can influence how a cell behaves. Cell culture, another fundamental concept, involves growing cells under controlled conditions. This technique allows scientists to study the physiology of cells and how they respond to various stimuli. It's like having a microscopic lab where scientists can experiment with life on a cellular level. But what about the tools of the trade? Techniques like PCR or polymerase chain reaction have become instrumental in biotechnology. PCR is a method used to amplify a single copy or a few copies of a piece of DNA across several orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. And then there's bioinformatics, the use of computational tools to manage and analyze biological data. This field has exploded with the advent of big data and has become essential in analyzing and interpreting genetic data. However, with great power comes great responsibility. Ethical considerations in biotechnology are paramount. Responsible research and applications are key to ensuring biotechnology benefits humanity without causing harm. So, to recap, biotechnology involves manipulating life at the molecular and cellular levels. It's about understanding and tweaking genetic engineering, recombinant DNA technology, DNA sequencing, protein expression, and cell culture. Tools like PCR and bioinformatics are essential, as is the importance of ethical research and applications. The world of biotechnology is complex and constantly evolving, but its potential is limitless. It's a field where we have the power to shape life itself, and that is truly an awe-inspiring thought.